Okay, Chapter 7. We're going to talk about how energy gets released from the food we eat, basically glucose we'll be talking about, and then how it gets transferred as a energy currency in the cell, it's basically ATP. So here we go. Woo, zooming in. Okay. Um, Skipping ahead. All right, here we go. So, the next chapter we'll talk about how light energy is captured by plants, the primary producers, in basically chloroplasts. We'll focus on those in the next chapter. And that energy is captured and it produces organic molecules and oxygen. And basically those organic molecules and oxygen are what are going to feed into the process we'll talk about in this chapter, which is cellular respiration. We'll talk about two different parts of it. Glycolysis that happens to the glucose initially in the cytoplasm, and then the electron transport chain and citric acid cycle that happen inside the mitochondria to produce the most amount of ATP. And basically the products of those generally are CO2 and water, and that basically goes back into the ecosystem for the plants to use again, so we have a nice circle. Before we get into what happens to glucose, we have to talk a little bit about energy movement. And you might remember, I hope you remember from chemistry, the words redox, reduction, and oxidation. Reduction means a reduction in the oxidation number, which is that charge. And oxidation is basically losing electrons. So there's this name, oil rig, which you can remember easier. Oxidation is loss of electrons. Reduction is gain of electrons. Because remember, in chemistry, when we talk about bonding, talk about any of that, we're really talking about electrons interacting or being shared or actually moving. An easy way to think about reduction would be a sodium ion. Remember Na? It tends to lose an electron, so it gets oxidized. Oxidation is loss of electrons. And then chlorine would be gaining it, so it gets reduced. And that's a full-on movement. Those are really easy. We'll look at another case that's pretty easy. But let's look at a harder one. Your book talks about it ever so briefly. We're still talking about where the electrons are and how closely they're held to a molecule. But now we're talking about the fact that energy is involved to move the electrons. And this is a little bit craftier example here. So methane, remember the carbon and the hydrogen tend to share the electrons almost equally. Oxygen here is going to share its electrons exactly equally because it's a diatomic. So what they're saying here is that the carbon is going to become oxidized. So remember, oxidation is loss. It's going to lose its feeling of electrons. So here the carbon is sharing it equidistant to the hydrogen. Over here, remember, oxygen is a lot more electronegative. It's actually hanging on to those electrons more. You can see it also here, hanging on to its electrons more, whereas the carbon is feeling them less. So energy has been taken to move those electrons this way. And this is sort of a more subtle way of thinking about oxidation and reduction. So in this case, the carbon has been oxidized and the oxygen has been reduced. Don't dwell on that too much. Let's look at the example that we need to focus on for moving energy around in the systems that we'll be talking about. So in ripping apart your glucose, you're going to be breaking bonds to release energy. And we want to capture that energy in another chemical form to be able to move it around places. And the guy we're going to use to move things around, sorry about the helicopter noise here, is NAD. And here it's called NAD+, just like school. Watch that nitrogen. You also are going to recognize some other functional groups and molecules we've talked about before. So here's the A, here's adenine, here's a ribose, it's got two oxygens, two phosphates, hooked up to another ribose, and then this nicotinamide group is up here. So the nicotinamide, my goodness, we just have everybody here, is going to actually gain some electrons. It's going to go from being a positive charge to neutral, which means that it's going to get some electrons. And then look up here, this hydrogen, we're actually going to gain a full hydrogen. But along with that full hydrogen, we're going to get an electron with it. So this molecule here will be reduced. It's going to get electrons and move them around. So nicotinamide, positive charge. We get 2 hydrogens from the food we eat floating around in our bodies, moving things around. And let's separate them into 2 electrons and 2 protons, basically 2 hydrogen ions. Basically, that's what that guy is. Enzymes are going to be involved, ripping that apart, and when bonds break. And then 2 electrons will go. We're going to satisfy that nitrogen with 1 of the electrons, and then that 1 electron plus that proton will become part of this guy. And we basically have gained 2 electrons, and we can move that energy around. In the process of it, we make 2 hydrogen ion protons, which we're going to see get pumped around and do some work for us later. Oops, I did have a zoom in on that. So again, NAD+, we'll be talking about that a lot, gains electrons, gets reduced, and we're going to bring it around to do other things. So why do we need to do that? Well, we need to slowly, wow, I didn't want you to do that, slowly siphon off the energy from the reactions we're going to do. We know that we can burn things, we can have an explosion, and you get this huge release of energy, but your cells can't do that. We need to basically save that energy and move it around in a place to do more work for us. Remember, we want that free energy to do work. So what we're going to do is basically use this thing called an electron transport chain, which they have as these little propellers. We'll talk about what they exactly are later. That the electrons are going to slowly bounce down to molecules of higher of different energy states, and then in the process we'll make ATP. So this is cellular respiration. Okay, where does cellular respiration take place? Well, imagine this whole screen here is a cell, and that this like light tan area is the cytoplasm, and then this is the mitochondria. Mitochondria, or organelles, we've already heard about, with these nice inner membranes, lots of extra surface area. We'll be talking about that. So first we'll take apart glycolysis. We're going to bring glucose in. We're going to go through a series of 10 enzyme steps, make a little bit of ATP, make this molecule called pyruvate, also make a little bit of NADH. So remember, we reduce NAD to become NADH. Then we'll bring the parts of that pyruvate molecule into, well, all of it in, but only part of it will then enter the citric acid cycle. We'll bring that across the membrane of the mitochondria. That 2 carbons will enter the citric acid cycle. Again, break bonds, rearrange bonds, make some energy carriers, feed that into the electron transport chain, which is embedded on this inner membrane. Again, we're going to see lots of pictures of that in a little while, and make a ton of ATP. The substrate level ATP production, I'll show you a picture of that, means that an enzyme is physically grabbing a phosphate and adding it to an ADP. Not terribly efficient. We make a few ATP here. We make a lot of ATP in this process here.